let's talk about what happens at a synapse. Now, this occurs whenever we have a signal traveling down one neuron and then going to another neuron, to another nerve cell. So this is how the message gets from one neuron to the next. So in this case, we'd have the signal going in this direction. Okay? So this is the axon terminal of the first neuron, and this is the dendrite, the part that receives the signal on the second neuron. Okay? Notice that the two neurons do not touch. Okay? There's a space between the two neurons. That space is called a synapse. And we're going to take a look at the events that happen at a synapse. Basically, that signal has to get from the first axon to the second axon, right? And it's got to go across this gap. So how does that signal jump the gap? Well, quite simple. When the action potential gets to the end of the first neuron, it will cause the release of a chemical. So you can see we've got these chemicals stored in these vesicles. When the action potential gets to the end, it's going to cause these vesicles to fuse with the membrane by exocytosis, if you remember what that is. Basically, this chemical is going to be released into the gap. Now, this chemical is called a neurotransmitter, and it's called that because it transmits signals between two neurons. It's a neurotransmitter. The most common neurotransmitter is ACH, which is short for acetylcholine. Now, there are others, but that is a very common one. So quite simply, when the action potential gets to the end of the first neuron, it causes the release of the neurotransmitter. So that neurotransmitter spills out into the gap. It's going to diffuse across this space, and when it gets to special receptors on the second neuron, on the dendrite, it will bind, and when it binds, it causes the opening up of sodium channels. And you remember when sodium channels open, sodium ions flow in, and that leads to an action potential. Okay, that's great. So now I've got all these neurotransmitter particles attached to these receptors, and that's causing a continuous influx of sodium. Well, this dendrite can never get back to resting potential. It can't be used again unless it's allowed to get back to resting potential, right? If all of these neurotransmitters are just allowed to stay in this gap and attached to these receptors, it's going to cause this dendrite, this neuron, to be continuously separated. So we need to sort of turn this off so it can get back to resting potential. So we need to remove all of these neurotransmitter particles from the synapse so that this neuron can reset itself, okay, can go back to no reaction. And that's the job of this enzyme that destroys the neurotransmitter. Released into the gap, we've got an enzyme that selectively attaches to the neurotransmitter and breaks it apart. And when it does that, it will detach, cause a detachment of all of these neurotransmitters, and that will allow this neuron to go back to resting potential. So if the most common neurotransmitter is ACH, the enzyme that breaks down ACH we can call ACHase, or the long name would be cholinesterase. Okay, so let's take a look at a short video here. Kind of illustrates what's going on. Okay, so let's go down here. Yes. Okay. Next. Okay, so here I have an axon, okay, and here's indicating this signal being transferred down the axon of the neuron from the dendrites to the axon terminal. When it gets to the end, okay, when it gets to the end of the axon terminal, it causes the inrush of calcium ions. Now, this is not something you need to know for Bio 30, but the result of that is when the calcium rushes in, it's going to cause those vesicles to release their contents, to release the neurotransmitter into the gap. So the neurotransmitter then enters the synapse where it can diffuse across and contact the dendrites of the next neuron. And when it does that, so here's the neurotransmitter, here's the receptor. When it does that, it causes sodium channels to open, and when sodium floods into the cell, that causes an action potential in the next neuron.